and environmental situations. So now I'd like to introduce Professor Robertson briefly. He is a dean of the College of Engineering, University of Wisconsin-Madison, US. He joined University of Wisconsin in 2013. Prior to this, he was a Donald B. Willett Professor of Engineering in the Department of Material Science and Engineering at the University of Illinois at Urbana-Champaign, and served as head of the department from 2005 to 2009. Dr. Robertson served as the director of the Division of Materials Research at the National Science Foundation. Lastly, I'd like to add one more career to his history. He was one of the principal investigators of I²CNER until 2019. His insight to the mechanisms responsible for hydrogen embrittlement of metals was recognized by the Department of Energy in 1984 when he received the DOE Prize for Outstanding Scientific Accomplishment in metallurgy and ceramics. In 2011, he received the DOE EE Fuel Cell Program Award for contributions to our understanding of mechanisms of hydrogen embrittlement. He was selected as the 2014 recipient of the ASM Edward DeMille Campbell Memorial Lectureship. For more detail, please look at flyer. Professor Robertson, could you start presentation?
Yes, let me just see if I can share my screen with everyone.
Okay.
So hopefully everyone is able to see the screen. Petros, you can see the screen?
Yes, clearly. Yes,
all right,
clearly.
Good. Thank you. So let me begin by wishing all of you a good morning and also asking after you and your families. These are very interesting times that we find ourselves working in, and certainly very different ways that we normally collaborate. And certainly, I never thought we'd be using Zoom or any of these tools quite as much as a way for us to talk to each other. I still prefer meeting in person and being able to listen to your ideas and hearing from you. So what Petros asked me to do tonight is to talk to you about how hydrogen impacts the evolution of the microstructure and what we can learn from it. I was a little bit embarrassed when Professor Kubota was reading out my introduction, because it also said something in 1984 certainly dates how old you actually are. So that was a little bit embarrassing, but hopefully you'll get something from this. I will say that for those of you that are still working in the area of hydrogen embrittlement, this is just a fascinating time to be working in any of these new areas. You have so many new tools and capabilities, whether it's experimental tools, whether it's computational tools, that you can actually bring to bear on the problem. And I hope that the talk this evening will make you ask some questions about what tools should you be using to probe how hydrogen is causing materials to fail prematurely. I'd encourage you, and hopefully this talk will do that, not to stay with the tried and true methods of exploring hydrogen embrittlement, but to take advantage of all the wonderful new tools that are out there and apply them to this problem, because I believe there's new insight there to be had, and you're the ones that are going to be able to do that. So let me start the presentation by just sort of talking you through what happens with a simple stress strain curve in a metal system. The system really isn't important as long as it's not a hydride forming system. So this talk will feature nickel, stainless steels, and some other materials, but the material isn't really the important part. The important part is the development of the knowledge to try and explain hydrogen embrittlement. So I'm sure many of you have seen simple engineering stress strain curves. You'll see that the material will start to plastically deform. It goes up, reaches some maximum in its tensile strength, and then it will sort of start necking down and will ultimately fail. The properties are dependent on temperature. They're dependent on strain rate. This just happens to show one particular example where we're comparing the effect of temperature on the stress strain rate or the stress strain curve. But what I want to do is actually take you inside the material and ask, what gives rise to this macroscopic property? What's actually going on inside the material to give you that response? So hopefully I'm going to show you a couple of videos here and that these are going to work. For those of you that aren't used to looking at electron micrographs, let me explain first what you're looking at. You'll see some sort of dark lines on the screen. I guess there's several dark lines, but the ones with the sort of black-white contrast, those are the dislocations that exist inside your material. These run from the top to the bottom of the electron transparent foil. In both images, roughly in the center of the image, you'll see a darker feature. And I don't know if I move my mouse, you will see it, but this feature where the hand is, that's actually a precipitate that sits inside the material. So what I want to do now is actually play these two video clips for you. And what I want you to do is focus on the dislocations that are actually around the precipitate, and for you to watch how the dislocations break away and move past that precipitate, right? The precipitate's an obstacle, and we're going to see how the dislocation goes past. And this is what's happening inside a material when you apply a load to a bulk specimen. So let me see if the first one will play. And you can start to see the dislocation to move. You can see one creeping up on behind it, and then it suddenly just breaks away from the particle. And it would just keep doing this for some period of time. Now I'm going to show you the second video clip. It's the same particle, same dislocation types, but hopefully what you're seeing is that the dislocation is now pinned longer at the obstacle and is in fact breaking past the obstacle in a different way. So the question you should be asking yourself, if the precipitate didn't change, if the dislocations didn't change, what was responsible for the change in the interaction? So what's responsible for the change in the interaction is one dislocation interacts with the precipitate, it passes the precipitate, but it leaves something behind. So though we cannot see the change in the precipitate form does change as more and more dislocations go past it. And you actually require a different mechanism for the dislocations to bypass with increasing strain. So my first message I want to leave you with is, just because we might understand what happens when a material initially deforms, that interaction does not stay the same. Things change inside the material as the dislocations move and they interact with other precipitates. They're going to change when they interact with grain boundaries and with each other. So just because we understand dislocation mobility at the very early stages doesn't mean we understand them as a function of strain. So what happens is we keep applying more load to these. What happens to the dislocation structures that you see? So here's just an example. It happens to be a nickel. The numbers that you see, the 8.5, the 9, and the 26, that tells you the percentage of plastic strain that this material has been subjected to. You're not looking at individuals' dislocations now, but you're looking at collections of dislocations. And what you'll notice again is you'll see this collection of dislocation here. You'll see also over in this area, the dislocations have now started to self-organize into a structure. We call that a cell structure. And you'll notice that at 8.5% strain, you get the beginnings of a cell structure, but it's not well formed. If you go to 9% strain, if we could actually look inside these sort of thick black lines that you see, we would actually find individual dislocations all sort of bunched together in that dislocation cell wall. And you can see some of them are different orientations. The cell boundaries are making cells of different size. And as you keep straining, you will actually refine the grains, this cell size to smaller and smaller sizes. And that's what you see at 26% strain. You'll actually see much smaller cells. Of course, ultimately, what will happen is the material will get to a certain cell size and then it will stop, right? The material can no longer accommodate the plastic strain through dislocation cells and the motion of these dislocations between the cell walls. Other things will have to start happening. So what starts happening is that you get rotations between the individual cells. And if we look further, what happens, this is what you get. This is what happens when you cold roll to 90%. This is the kind of structure that you would see in your material. You'll now see that you've got different types of boundaries, so geometrically necessary boundaries, and they define regions of your material. And between those boundaries, you've now got other types of cell walls. They've called them IDBs in this as another type of boundary, of dislocation boundary. But this is what exists in your material as you deform it. Now, if you think of now moving a dislocation in this structure, it would be very, very different than one I showed you at the beginning in the little movie, right? It's just, the microstructure is more complicated. There's more barriers to the dislocation motion. And the more barriers there are, the more stress, more load you're going to have to apply in order to get the dislocations to move. So this is what happens just in a metal, no hydrogen, just loading up the material. So this is the kind of work I used to do. And at the time, I would say, at least I hope we'd have said that it was advanced for its time and we were actually forging new grounds in some of the things we did. But today, you can actually go much, much further. And you can then say, okay, can I actually extract quantitative information from those microstructures that I showed you? And the answer is, yes, I can. I can actually derive more information that I can then relate back to the macroscopic stress strain properties than I used to be in be able to do in 1984. So what kind of information can you get? Well, here's just an example. This happens to be a low carbon steel that was deformed under cyclic loading. And in this case, the macroscopic specimen, we stopped the loading before the crack had propagated all the way through. So it's an arrested crack. We then took samples at different distances, at 10 microns, at 33 microns, and 56 microns from the crack tip. And we said, okay, what does the microstructure look like at those distances from that crack? And you see the difference, right? You can see sort of very detailed, smaller cell structure at 10 microns. You still have a cell structure at 33 microns, and you still have one at 56 microns, but the cell size is much bigger at 56 than it is at 10. And at 56, you can get the idea that there's still individual dislocations within the cell wall. And this is what you would expect, right? More deformation close to the crack tip. You're seeing that in the smaller dislocation cell size. Now, one of the things that you can do, and this is all the work of Shui Wang, so Shui should certainly get the credit for doing this very, very elegant work. Because what he then did is he said, okay, if I measure the cell size, I can actually relate that microstructure I see to a local flow stress. Right? So you could calculate the flow stress related to the microstructure from knowledge of the dislocation cell size. And that's what you see over on the right-hand side. You'll see that as you move with increasing distance from the crack tip, you'll see that the flow stress decreases. Now you'll say, well, we knew that, and you're absolutely right, we knew that. We knew that's exactly what you would predict. But this is a case where we've actually now come in and said experimentally, let's see if we can get that information, and is the result reasonable? And I would say, yes, the result is very reasonable. It's about the right order of magnitude of what you would expect. The shape of the curve is about right. And you can do this in multiple different samples. So we can actually get ideas about the magnitude of the stress as it approaches the crack tip from knowledge of the microstructure. And I'd say that's a new thing that we can do today that we couldn't do before. So that's one thing I want you also to bear in mind. We can do other things now too. As I said, as we increase the level of deformation and we make the dislocation structures tighter and tighter, those cell structures get smaller and smaller, the next thing that must happen is you'll get local rotations. Just think of trying to deform something. It's going to try and deform. It can also rotate to take into account that deformation. Each of the cells will rotate slightly differently. It makes perfect sense when you think that's what's going to happen inside a material. But can you measure it? And today you actually have systems that you can put inside your electron microscope that allow you to make EBSD maps using the probe of the electron beam. So now you're talking about a beam size that's much smaller than you'll get in an SEM, even in your best SEM. It's much smaller. And you can actually start pinpointing where the stress is coming from. You can actually measure it. So the two color-coded squares that you see here are just two areas of a grain. And we've gone in and we've measured the misorientation. And you can see the difference. Now, the way we've plotted it in this case is we've plotted it with respect to the reference point. And the reference point is actually this white circle, or this black circle with the white border that you see up at the top. Everything is done with respect to that white dot. So what you will actually see is this area down here. If you were to come from the top to almost the bottom, you're seeing a local rotation of 10 degrees. If you go just less than a micron over, you're maybe at 5 degrees. And this is going to become important because this is getting us back to this idea of, can we measure the local strain inside a material? And can we use that information to give us insight into the mechanisms of hydrogen embrittlement? Right? But it's another technique we are now saying we are going to apply to try and understand what's happening in the material as we deform it. So this is new information that you can actually get. Let me turn to an example where we have put hydrogen in and just ask what happens to the material when we put hydrogen in it. Again, a very classic example. It's just putting hydrogen into pure nickel and looking at how putting hydrogen into the nickel impacts the engineering stress versus the engineering strain curve. And all of you have studied hydrogen embrittlement. You'd say, well, this is absolutely obvious. You're going to get a reduction in the ductility. You'll see that there's a higher flow, a strength on this with the presence of hydrogen, and it fails quicker. And, nickel is kind of nice because to accompany that reduction in the ductility, you also get a change in the fracture mode. So it makes it nice and easy to say that there is a big difference and there's been a different failure mode. So what you see here is that in the SEM images, in the absence of hydrogen, you get the distal type fracture that we're all very familiar with. When you actually put the hydrogen present, you actually get intergranular failure. And you can see that in the upper images, you can see the grain boundaries quite nicely. So this is what happens when we put hydrogen into the system. Now, one thing to remember, you don't always get a change in fracture mode where it will go from ductile transgranular to an intergranular failure when you put hydrogen in. It depends on how much hydrogen you actually put in your system. So just to show that, here's some old work of Dave Lasilla's out of the University of Illinois, where he couldn't measure how much hydrogen was actually on the grain boundary, but he could put different amounts of hydrogen into the specimen. And he said he made the assumption that if he leaves the sample at room temperature long enough, you'll get diffusion of that hydrogen and you'll get more on the grain boundary. And what he's showing here is that the percentage of intergranular failure will increase as you add more hydrogen into the bulk of the material. He also showed one of the other complicating things about hydrogen embrittlement as we try and understand it, and understand its effect on the grain boundary. If you actually add sulfur to the grain boundary, along with the hydrogen, the two act synergistically, and you only need a little bit of sulfur on the boundary, a little bit of hydrogen, and you'll get much more intergranular failure than you do if you have a pure boundary. So as you think about hydrogen embrittlement mechanisms and how you could possibly get a transition from transgranular to intergranular, you also have to take into account this impurity effect. So any mechanism you come up with has to be consistent with all of the experimental evidence. And this is another point I want to leave you with tonight, is that don't just interpret one experiment and say, well, this is the mechanism for hydrogen embrittlement under this case. Put all of the information together and ask the important question, is the mechanism I'm proposing, is it fully consistent with all of the information I have, not only from your lab and your experiments, but from the work that's in the literature? You don't actually have to believe the explanation in the literature. Use the data and say, can my mechanism, can it explain all of these things? Because I think all of us would agree that if there's a different mechanism for every material and for every condition under which we test it, we're never going to solve the problem of hydrogen embrittlement. We're just not. It's way too complicated. We have to look for commonalities to find that general explanation. So I said at the beginning, one of the things I think that you're incredibly unfortunate in being this time is that you have all sorts of different tools at hand that you can apply to the problem. So this is another work from Shuai, and he has been using as this grand canonical thermodynamic model to actually look at what's the reduction in the cohesive strength of a grain boundary due to the presence of hydrogen. And you can see his results here on the left. You pump hydrogen onto the grain boundary, you do decrease the cohesive strength. If you put enough on there, you'll decrease it a lot, and yes, it will fail. The question that comes back to you to ask, though, at the very extreme of this graph where it says, yes, the boundaries are incredibly weak, is it practical under the charging conditions that you use in your lab to ever put that much hydrogen into a material and into the grain boundary? Right? We all know the experimental conditions under which we charge microscopic specimens. So if you're looking at this and say, well, yeah, it's going to fail, go back and ask the question, is it physically possible for me to put that much hydrogen into the system and onto the grain boundary in order to get that magnitude of decrease? The other thing that Shuai's work shows is that the amount of reduction that you get is grain boundary dependent, which I think is it's sort of obvious, but it's quite fascinating. So it says that even if you pump in X amount of hydrogen, and that would cause one boundary to fail, would that lead to system failure? Because all the other boundaries are strong and the crack won't propagate along the boundaries. So you need enough hydrogen on enough different types of boundaries in order to be able to get intergranular failure across the entire specimen. Now, what I would really like one of you to do is to take samples, not just one, but many samples. Can you actually explore the grain boundary orientation, start looking for where the grain boundaries are going to fail, and do something like Shuai's done and say, let me now calculate how much hydrogen is there and what's the magnitude of the reduction in the cohesive energy. And then ask the important question, is it enough hydrogen, enough reduction on enough grain boundaries in order to cause the material to fail just by splitting the boundary itself? So I'd actually like to go do that experiment, but my other job actually stops me doing that. But hopefully one of you will. And when you complete it, send me your paper because I'd be fascinated to read it. And it's okay if you come back and say, what you proposed and what you told us is completely wrong. Here's the right answer. I'm okay with that as an answer too. And I'd love to talk to you about doing that experiment. So I just mentioned that the grain boundary could fail, and that's been one of the classic mechanisms that's been put out there. It's the hydrogen-induced decohesion mechanism. What it says is that it really, hydrogen goes on the grain boundary, reduces the boundary cohesive strength enough that it just fails. Any plasticity that happens in the material is totally irrelevant to the failure. There's nothing to do with it. Is that right? We also know that hydrogen transports, or dislocations transport hydrogen. There's some very elegant experiments that have been done that show that. So if hydrogen is transported by the movement of dislocations, and we need to put enough hydrogen on the grain boundary to get decohesion-induced failure, is it right for us to say that plasticity has no role in hydrogen-induced decohesion of a boundary? I'd say, no, you need to pump more and more hydrogen on the boundary, so that transport is important. The challenge with that mechanism, though, if you've ever looked, and I didn't show you this evening, but if you look at dislocations hitting a grain boundary, they'll go into the boundary, but the grain boundary will also emit dislocations. If that happens, the dislocations go in, bring in hydrogen. The dislocations going out, they take away hydrogen. So remember, one of the things I asked you to think about is that if we're going to make the assumption that it's just straight hydrogen concentration in the boundary that causes decohesion, can we make the assumption, or is it right for us to make the assumption, that the hydrogen concentration is static while you've got plastic processes? And I would say, at least in the early stages of straining, that can't be true because dislocations do move hydrogen. So now we've gone from a static hydrogen concentration on the boundary to a dynamic one. And I don't think anyone's ever thought about that type of interaction with a grain boundary. And again, it's one of the challenges I'll put to you, especially to our young scientists who are here tonight. I'd love to see you try that kind of experiment and think about it, or the simulation. And I'd love to see the results in that. And again, I don't mind if you tell me I've got it completely wrong. The other thing I said is that it's very, very important for you to say, let me look at all of the results that are available and see if my mechanism holds up. So one of the things I showed you earlier was that at 77 Kelvin, you get a significant reduction in the ductility. You get a change in failure mode. Now, if we're really working at 77 Kelvin, we know that hydrogen is going to be trapped in the lattice. It's not going to go to the dislocations. It's going to act as a solid solution strengthening agent. It also says that if we're not moving hydrogen by dislocations, and that the diffusion through the lattice or along the grain boundary is low because of the temperature, it says that the starting hydrogen concentration is the one that's important for inducing the decohesion. Right? That to me, that's what that result tells me. I'm going to see if I can go back in slides, which is something I normally don't do in a talk. But let me go back and show you this. And here's the question I want you to think about. At 77 Kelvin, we said that all of the hydrogen should be on the boundary because there's very little diffusion that's going to happen either through the lattice, along the grain boundary, and there's going to be very little transport by dislocations. So the hydrogen concentration that's on the boundary at the beginning is the one that caused the decohesion. If that's correct, if you look at this stress strain curve, why did we need to load it up to over 60%? I find it fascinating that we would say that all of that deformation that's happening, all of that accumulation has had no impact on the mechanical properties. But that's what the classic decohesion explanation would tell you. So I would ask you to think, is that right? So now what I want to do is to take you more to what I think is happening, and again, to pose some questions to you. Professor Kubota was very kind when he talked about my 1984 work and the fact that it led to some recognition from the Department of Energy. So this is, in fact, one of those early movies that we made about adding hydrogen into an electron microscope, applying some load, and seeing what would happen to the dislocations. In this particular example, we applied some load. We knew these dislocations were mobile. We took the load off the specimen and then simply added hydrogen gas into the electron microscope. And we allowed that hydrogen to interact with the metal. Now, one of the things that does happen is that the hydrogen electron beam comes down, interacts with the hydrogen molecule. So you'll get atomic hydrogen. So we have high fugacities present at the sample surface. So I think we're pretty confident that we were injecting hydrogen into this sample. So what happens when we inject? So you'll see the image degrade slightly, and then you'll see those long black lines just start moving. There is no increase in the applied load. It's all due to the presence of the gas in the sample. We could take the hydrogen back out, and the dislocations will stop, which they should, because the hydrogen embrittlement effect is in fact reversible, right? You take the hydrogen out of the material, you're going to go back to the old behavior and the old failure. You put the hydrogen back in, it should go back to the hydrogen embrittlement. We were able to show that by adding hydrogen into the microscope, taking hydrogen out, putting it back in. The dislocations would always start moving. Now, I enjoy following the literature, and I've seen some reports recently that said, well, they did the experiment wrong because the fact that the dislocations move was all due to the pressure differential of introducing the gas into the cell. You know, we did think of that as a possible mechanism. So what we did is actually we introduced argon gas into the microscope. We put other gases in as well, but argon was easy to do. There was no effect. So then we had the very clever idea that why don't we use argon gas as a carrier gas, but before it enters the microscope, let's fully saturate it with water vapor. So of course we bubbled hydrogen gas through a bubbler full of water into the microscope column. I wouldn't say it was my best experiment ever, because putting water vapor or water into an electron microscope column doesn't make you any friends with the rest of the microscopists, but that's what we did. So what happened? Well, when we put in the water saturated gas, the dislocation started moving. We took the water saturated gas out, they stopped moving. We put in the dry gas, no effect. I'd have said there should be some pressure differential effect. So if that was the reason for the dislocations moving, they should have always moved when we put in any gas, and they didn't. It was only when there was hydrogen or water vapor present. The other thing I would say is that we actually did these types of experiments. I think any material that you could think of that's embrittled by hydrogen, which is almost all of them, we tried this experiment on, and it worked on every one of them. It was a universal effect. It wasn't just happening in nickel. It wasn't just happening in a stainless steel. It was universal. Well, you might ask, has anyone ever repeated your experiment? The answer is yes. There are reports out of Sandia National Labs where they did put hydrogen into their microscope. They put enough pressure in so that they actually got enough hydrogen going into the system, and they see the same things. The dislocations would move. The other thing we did to actually show that these were the right kind of experiments to be doing is we did put titanium in. And we get a beautiful hydride field emanating from the crack tip that we produced. Beyond those hydrides, we actually saw the dislocations move at an accelerated rate because of the hydrogen. So you saw both effects happening in the same material. So could it be a pressure differential? I'd say no. I think we did enough experiments to go against this. These experiments were also done in a high voltage microscope, which means they were done in relatively thick specimens. I mean, relatively thick for an electron microscopist. So they're still thin, they're still electron transparent, but thick by our standards. Some of the other things to show you. So one of the things that comes up, we've said that hydrogen's going to accelerate the mobility of dislocations. At least that's how we interpreted the microscope experiments we did. If that's true, one of the things that we would predict is that for the same level of plastic strain and material, we should see a different dislocation microstructure in the absence and the presence of hydrogen. So again, this is work done by I²CNER, looking at nickel following high pressure torsional processing. The upper images show the uncharged material, and the lower one shows the hydrogen charged material at two different plastic strains. Now, the beauty of the torsional processing was that the materials were getting the same plastic strain. And by picking the same locations on the samples, we could say that those samples had experienced the same plastic strain. If you look at the upper image on the left, you'll see a dislocation structure, but you'll also notice that there's areas where the dislocation structure is poorly formed, and some of the cells are actually really quite large. If you compare that to the bottom image, one of the things that you'll find is that the cell size is now much smaller. And remember, cell sizes say that the processes have gone further. In the old days, we'd have said that means the material's been subjected to higher plastic strain. They can't be in this case because the experiment was done to yield the same level of a measurable plastic strain. Right? So we've just negated one of the arguments that says these microstructures, the difference, you've taken them at different plastic strains. They aren't. The experiment was designed to give you the same plastic strain, so the microstructures are different. So why is that important to point out to you? Well, one of the things that it should tell you is that we've made assumptions before about how dislocation microstructures or how plastic strain evolves in hydrogen in the absence of hydrogen, and we've said they're the same. They can't be, because clearly one is moving faster than the other. So do we, as we do the modeling that Petros likes to do, is he using the right expressions, the right formula in the presence and absence of hydrogen? Or is there still work for him to do to actually find out why it's getting this accelerated structure? It's not the same correlation between microstructure and strain. There's something different in hydrogen. And I challenge Petros and others in this area to say, do we have the right model yet for putting the hydrogen effect into those models? And I don't believe we do, because we've always based it on our past understanding. Petros probably isn't going to like me for saying that, but I think he still has work to do. And for those of you that are working in this area, I look forward to you again showing me that this idea is either right or wrong. So as we go up and we say, well, can we see it in more than one material? This again is an example from nickel. We're looking at the same level of strain. And I must admit, now looking at this figure, I forgot to put the grain orientation on the sample. You know, one of the challenges in electron microscopy that we face is people can always say, well, the microstructure that you see depends on the grain orientation. And they're absolutely right. You must compare microstructures in the same grain orientation. So one of the beauties that you have today is that you can do EBSD analysis in a sample. You can identify the grains and the grain orientations prior to deformation, because those orientations will change when you deform, but you can measure it prior, identify those same grains later and say, well, I know this grain started as near 001 or 001, or sorry, 011. So you can pick that starting orientation and you can compare them. And that's what we've done in these three images here. So you'll see a comparison at 8% strain, comparison at 9.5. In this case, the hydrogen charged one failed at 15%, and the other one didn't fail. It was still up at 26% strain. And if you look at the microstructures that I've been trying to get you to look at them, 8% strain, no hydrogen, a sort of ill-defined cell structure. If I look at 8% with hydrogen, still the early stages of cell formation, but more formed than the uncharged. Right? Again, it goes to this accelerating effect. If you look at 9.5 uncharged, you'll see, yeah, you're starting to see a cell structure in patches, but it's not uniform. 9.5 in the presence of hydrogen, it's pretty uniform. And certainly, if you look at 15% strain, if you were just to put these samples down in front of anyone, my guess most people would say that the one I've labeled 15% strain actually had the 26% strain because it looks more evolved. It is more evolved, but it's got hydrogen there. But this is the kind of information you can get today. So one of the things I told you can do is you can start to think about imaging the local strain. You can get information from the microstructure. I've shown you how to do that, and I'll take you back to that in a moment. But you can also visualize it. So I said earlier that you could do EBSD on an electron microscope specimen using the beam size of an electron microscope. So the one example you see here is, again, nickel. It's hydrogen charged, it's uncharged. The hydrogen charged is 15% strain. The uncharged is 26% strain. The white line that you see roughly in the center indicates a grain boundary. The black dots with the white borders, those are our reference points. And we take a reference point so we don't have to worry about the misorientation difference as you go across a grain boundary. That's been neutralized by taking these reference points. What do you see? If I look at the two sets of images, I notice that in the upper one, there's no real strain mismatch or misorientation mismatch because it's a dark red, a light red on both sides of the grain boundary. What does that tell you? Well, if we think about deformation of a polycrystalline material, you have to get communication of strain across the grain boundary. The reason the boundaries stay intact is that you can communicate that strain across the grain boundary. It's not always done by the transfer of dislocations, but what's happening on one side of the boundary influences what's happening on the other. If it's a good, strong boundary, it will communicate the strain from one side to the other very well. If that's true, there should be no mismatch in the misorientation difference as you go across. And you'll notice there is no real difference here, at least in the upper image. Now I ask you to turn to the bottom image and look at the difference there. I do have a different microstructure, but one of the key things I want to point out to you here is look at the stress or the strain evolution on the left side of the grain boundary compared to the right side. And look in particular at the difference right next to that white line that we have, the grain boundary itself. You'll notice that it's dark red on one side, and it's a different shade of red, maybe even orange on the other. What does that tell you? Well, if we look at the scale bar, what it tells you, there's more strain on the left side than there is on the right. So what could that possibly mean? It says to me that the strain has not been well communicated across a grain boundary. Now, if we think of taking an interface, which a grain boundary is, and we can't transfer the strain across the boundary, but you're still applying load, what's left for the system to do? It has to find some relief somewhere. And I would sort of put to you that it's going to cause cracking of the grain boundary, because now you've set up an incompatibility constraint across the grain boundary. And I've just shown you evidence that says there is a difference in these boundaries. And I could show you many more examples of this. It's not just a one-off. So there is a difference. What would cause the difference? It's not the hydrogen on the boundary. So what could cause it? Well, remember, I've shown you that the microstructure on either side of that boundary in the presence of hydrogen is way more evolved than it is in the uncharged. If you also remember, I said that the cells, as we load up more and more, start rotating with respect to each other as they try and move to a higher order configuration. So you're developing more strain on one side. It should try normally and go and communicate across to the other side, but it can't. So you would get strain mismatches across this interface in the presence of hydrogen. So it's the microstructure evolution and its development that gets you to this point. We could go back to the earlier example I showed you from Schwei and said, can we calculate what the local stress is ahead of a crack tip due to the presence of hydrogen? And we were going to get this from just looking at the microstructure and looking at the cell size. So here's the comparison images. They're roughly at the same distance from the crack tip. Two different specimens, but roughly at the same distance. And you'll see there are differences in the microstructure. The one thing I want to point out to you here, though, is if I look at just the microstructure at 52 microns and 56 microns, there's no shrinking of plastic zone size. So again, raise the question, when you measure the plastic zone size using a large scale technique, is it sensitive enough in terms of spatial resolution to give you the information? Or at the scale level you're looking at, is your answer right, but you're not looking at a fine enough resolution? You should always challenge those questions and ask, are you looking at the right resolution to make that claim? Now, I did say that Schwei could actually come in and measure the flow stress as a distance from the crack tip. So the upper curve, the graph that you see, shows you the data of the flow stress as a function of distance from the crack tip. The red circles show you the one in the hydrogen charge case, the black squares, the one in the uncharged case. What does the data tell you? It says that the curves are roughly similar in uncharged and hydrogen charged, which is good because it says that the deformation processes, the dislocation interactions, they're not necessarily different in the presence of hydrogen and uncharged. And to me, that's a relief because I don't need two different processes for dislocations just due to hydrogen. They're the same. They're accelerated, but they're the same. What else does it tell you? Well, it says that the stress field that you're developing in the hydrogen charge case is actually higher than it is in the uncharged. I think that's an important experimental observation to make. So how does that explain things that we see in the macroscopic sense? Let's say it's acceleration of the cracking under a fatigue test in the presence of hydrogen. Can I explain that now that I have this information? I think so. So let me make this assumption. Let me say that there is a minimum stress ahead of a crack that I need to get in order to get crack propagation. I've picked that as the blue line here. Right, the dashed blue line, that's the value I've picked. You can pick any other value you want. You're going to come to the same conclusion. And I'm going to say, well, how far ahead of the crack tip will I get that level of stress? How far ahead will I experience it? And you'll notice that in the case of hydrogen, I generate that stress much further away from the crack or from the crack tip than I do in the absence of hydrogen. So does that say that the jump for each crack step, is it bigger in the presence of hydrogen because the stress to drive it extends further? I think it's a very interesting hypothesis for you to test. And again, I'm hoping that someone will do it and will show me this. But to me, this says that we can relate the crack propagation rate in the presence of hydrogen to the microstructure. We have a microstructural explanation for the enhancement in the presence of hydrogen. So if I go back to our original problem, can I actually explain why I get enhanced failure, why I get a switch in the nickel from being transgranular to being intergranular? So let me offer you an explanation. And again, this is something I hope you'll take me up on and you'll test me on it to actually see if this is right. So here's the thought that we have. We've shown you that mobile dislocations move. We've shown you that the motion is accelerated. Their generation is also accelerated. And we've shown you that the microstructure will evolve more quickly in the presence of hydrogen. If the microstructure evolves more quickly and you get smaller and smaller cell sizes, we've also shown you that local internal stress goes up when hydrogen is present. Right? We've shown you all of those things. We've shown and suggested that because of the microstructure, that small cell size, the strain that develops across an interface or across a grain boundary is not well communicated, and that the differences in the strain on one side of the boundary to the other gives you a compatibility constraint. And the only relief mechanism is for the grain boundary to then fail. That is now the weakest link in our system, and it may have been made the weakest link by the presence of hydrogen on that boundary, but it's been driven to fail because that's its last option for taking that extra applied load that you've put in your sample. So hopefully, as I've gone through the presentation, you'll have seen and hopefully encouraged to think about, can I start looking at microstructure? Can I then say, can I take from that microstructure, not just the observation that there's dislocations there, what extra information can I learn with the new techniques that are available to me? Can I take that and say, let me challenge the modeling community. Let me challenge the other community and say, this is what I see experimentally. How does that work within the framework you have? And can we learn from each other? So use new techniques, don't rely on the old ones. Hopefully this will encourage you to think broadly and try new techniques, bring them to bear on the problem, and hopefully solve this problem of hydrogen embrittlement. So my concluding remarks, as I see Professor Kubota is back, is that one of the things I've shown you, and you already know part of this, is that when we talk about hydrogen embrittlement, we'll often say that the material right at the beginning, the initial state of the material is important. I agree. It is really important how we process that material. And there's been beautiful work done at KU and at other places showing how that microstructure, that initial state, is really, really important. I've shown you that the microstructure evolves with strain. So I would ask you to ask the question, does that evolved microstructural state play a role in determining the failure mode? And I guess I've already made my other points. Use the new techniques that we have at hand to challenge whether you believe in decohesion, whether you believe in the help mechanism. Use those new techniques to challenge that thinking. And I look forward to someone telling me that they've found the answer to hydrogen embrittlement, and that some of those who like to make steels can say, we now know how to make a steel that we can put in a hydrogen environment and expect no embrittlement. So I'll stop there and take any questions, but thank you very much for your time and attention. Thank you.
Thank you very much, Professor Young Watson. So I'm excited by your lecture. I found drawbacks about seminar via online. It's difficult to send you applause through the screen. Sorry, but I expect many questions from audience. Actually, I saw many results observed by advanced instruments. Is there any question from audience? I saw Arno. Please go ahead.
Hello, good evening, Professor Robertson. Can you hear me?
Yes, I can.
Oh, good. Well, thank you for your very interesting talk. And I completely agree with you. I have a question. I think you mentioned it in your paper about hydrogen embrittlement understood, and you talk about this very briefly on the nickel alloy. It's the increase in flow stress. And in your paper, I think you said that at a specific strain and a strain rate and temperature, you would have a solute drag effect on the dislocations. And so that's basically my question is, there are more results now that I think that you have one by the group of Professor Koyama with 310S, and they said it's just solid solution strengthening. How do you not reconcile, but how do you explain the effect of solute hydrogen on this kind of increase of flow stress or yield stress?
So I think at the very beginning, right? So if you add hydrogen into your material, it's going to find different traps to go reside at. Some of those will be in solid solution, but we also know it will go to any precipitate that you have. Some precipitates are more effective than others. We know it will go to the grain boundaries, and we know it will go to any residual dislocations that you have there. So I think the hydrogen is there at all of these different sites. And so you have to ask is what happens when you apply a load and the dislocations start moving. Our argument would say that some of the hydrogen that's near dislocations or on the core of the dislocations will move with those dislocations, right? There are some very elegant works from very early on that shows that you can get, by applying load, you can actually get more hydrogen transport. And that can only be attributed to the dislocation. So I think that the hydrogen has to be attached to the dislocations in some way. And I think Petros has done calculations again a long time ago, where if you put hydrogen with any obstacle, it changes that obstacle stress field too.
Oh yes, yes.
The interaction energy between the dislocation and that obstacle with hydrogen is going to change. And I guess the part we often forget is that we always say it's reduced, but in some directions it's actually increased.
Yes.
Right. If you look at the change in the strain field interactions, they're different, right? So in some directions it's reduced, some it's actually increased. So I think it's all solid solution strengthening. I would say right at the very beginning, in the early stages of deformation, when you have dislocations moving as individuals, yeah, you're probably going to get some solid solution effects. But remember, very quickly you get into that cell structure formation.
Yes, yes.
Right? If you get in the cell structure formation, the dislocations, the distance they move is from cell wall to cell wall.
Yeah, okay, yeah.
And if you think of where all the hydrogen has to be then, how much is in solid solution when you've got a fully small cell size structure? Right? I'd argue that, and by the way, for those of you that like to try and use the atom probe to look for hydrogen, that to me is a key question, is how much hydrogen is in specific locations, and could you actually show how much hydrogen is on a cell wall versus the interior of a cell? And I think that's the sort of kind of information that I'm hoping to see come from some of the work that's being done today. because that would answer it, right? It says, is this hydrogen all trapped in the cell wall, or is it still in solid solution?
Yeah, that was basically, every time I've seen the increase of yield or flow stress with hydrogen, I was thinking, is it just that the test is too fast? The strain rate is it too fast, or is it something else? Or the paper by Professor Birnbaum about the localization and acceleration of competing. But now I completely forgot that, yeah, now you have the dislocation cells, and then
This is one of the things I'm really glad that you brought this up, because I think it's an important point I wanted to get tonight, is that, and I will admit, and for many years I'd have said our focus was entirely on individual dislocations and what hydrogen did to individual dislocations. And we forgot that when we actually load these materials up, dislocation interactions lead to tangles, they lead to nescent cells, then the cell size gets smaller and smaller. And if we just think of trapping densities, I would say there's an awful lot of hydrogen trapped in those cell walls, right? So I hopefully my thinking's evolved over time as we understand more about hydrogen. I'd love someone to answer the question of, does hydrogen influence the self-organization of dislocations as you're up at modest strains? How does it do that? I think, to me, that's a fascinating question for us to grapple with. I don't have an answer, by the way. I just think it's a fascinating question.
Yeah, thank you very much. I'm actually working on, I was talking about this last week about XRD to get the bulk evolution, but I'm having issues with twinning.
Yeah, we tried early on a few years ago to do some work at the synchrotron where we were trying to follow the evolution of the strain and a sample with and without hydrogen. We were still working at Cornell University trying the setup, trying to get the results, so we never moved it to one of the better facilities. To me, that's a fascinating experiment to try as well, because if we take the picture that strain, we always have this picture of strain just building up constantly. But if you look at some of the X-ray results out of Denmark, you would say, no, it doesn't, because you get pockets of strain coming in, you get disappearing, it appears elsewhere as the microstructure evolves. Doing that in the presence of hydrogen, fascinating experiment. I think it would be a great experiment if we could do it right. So hopefully you'll do that.
We are trying with cold rolling to have, how do you call, basically a strain control deformation.
Right.
But it's another kind of worms, because there's not so much work done in this kind of stuff, and the XRD itself needs a lot of, we need to check the anisotropy, which is worsened by hydrogen. So it's a process, but it's very interesting. And again, thank you very much for your talk today, because it's given me new hindsight and new way of thinking about the next steps.
One of the challenges we had with the X-ray work was that the models to extract the information were still being developed.
Yes.
And so we had these beautiful data sets, but were we confident that we were able to extract the information properly? And the answer was we weren't sure we could do it. But that problem will be solved with time, right? We will eventually be able to do that. And I think it's a beautiful experiment. I'd go back to a really simple material and not worry about the cold working. Even something as simple as nickel, just load it up, and is there a difference? I think it's just such an elegant experiment to try.
Yes, I agree.
Thank you for a nice discussion.
Thank you very much.
Is there any questions? Please raise hand if you have a question. So I have a question. I understood hydrogen reduces separation work of grain boundaries. But according to your results, hydrogen fugacity is extremely high. But actually, we have intergranular cracking. So is there any special mechanism that achieve such high hydrogen fugacity?
In the microscope experiments?
Yes.
Oh, yeah.
Please.
So in the microscope experiments, it's all attributed to the electron beam.
Yes.
So the electron beam will cause dissociation and ionization of the hydrogen molecule. And so that gives you atomic hydrogen right at the surface. We did an experiment on aluminum where we could actually measure the pressure because it was a bad specimen with a thick oxide layer. But it turns out the oxide was nice because when hydrogen went inside, we would actually blister the oxide. And from the blistering of the oxide and before it ruptured, we could get a measure of the internal pressure in that bubble. And so that was how we actually were able to measure and say that the actual fugacity was much higher than the external pressure that we were measuring. So that was the reason.
Okay, thank you very much. Good. Is there any other questions? Aki?
For me?
Yes, do you have questions?
Okay, so yeah, thank you very much for your nice presentation. So towards 2020, to achieve the carbon neutrality, so of course, hydrogen is the most important key. So now, what we have as an issue is material cost actually. So for example, 316L, low strength, no welding, high cost due to the chemical composition. So that's a problem we are having. And at the same time, actually, we are now in the transition to liquid hydrogen from gaseous hydrogen now. But now we are trying to use a lot of more liquid hydrogen. So so from the Brian's data, in the present hydrogen at 70K, that stress strain curve show the some effect in the presence of hydrogen, right? And but at the 20K liquid hydrogen, very low temperature, I don't think hydrogen has no effect. I don't think hydrogen has any effect on the material property, but it would be nice if we can hear your opinion about the usage of liquid hydrogen safely in terms of materials.
Aki, that's a really hard question. I think, Aki, the work that's, that earlier work from the summer day group where they changed the temperature, I mean, it's an interesting experiment, right? They did load it up. So I think it would be interesting to actually do some more testing to see what's the temperature dependence. And I think you're right. At really low temperatures where the hydrogen's not going to be moving and it purely is a solid solution strengthener at that point, I don't think it won't have the same detrimental effect. It will have another effect, right? And that's the equivalent of solid solution strengthening.
Yes.
So I would think at liquid hydrogen temperatures, it won't have the same embrittling effect that we see even at 77 Kelvin, where if you take something as simple as nickel, you get the switch to intergranular. I would think that's low enough in temperature, right? Because you don't get the same bonding to the dislocations, to the precipitates, to the grain boundary. So I think I would say at that temperature, you shouldn't have a problem. Your problem's going to be if you warm the sample up or the system up, right? What happens then? Right. So if it goes through temperature cycles, what happens then? Because during the cycling, presumably you'll load different parts of the material with hydrogen.
Yeah.
But certainly the operating temperature my guess is you'll be fine.
Mm-hmm. Yeah, I agree with you. Yeah, thank you.
Okay, thank you. So do you have any questions? I find Professor Yuhei Ogawa. Ogawa-kun, do you have a question?
Why
you
you have
there?
a question? What's your question?
Sure is giving a hand.
Sure, yes.
Yeah. Can you see me? Hi,
Yep.
Ian. Hello, Ian. Can you see me?
Hello. Yes, I can see you.
I cannot see myself, but okay, that's fine. Yeah. Thank you very much for your very nice presentation, as always. Yeah, it's very encouraging, I think, and I also see that you have a lot of expectation on the future work on this field. So one thing I think you didn't go further about the communication of the strands between those grains. So what is the physical reason hiding behind this? What is your expectation about some work we'll do to confirm this? I think we discussed this before, but yeah, I want to know your comments on this. Yeah.
So it's just why we have this in. So those observations are done in nickel. There's a little bit of work done in a high entropy alloy that shows the same sorts of things.
Yes.
So one of the things I didn't talk about is even before we get to that stage, but you have the capability today to actually measure the evolution of the strain on the sample surface. And does that change the presence of hydrogen? Professor Kubota, or no, Andre wants to do it in the bulk. But if you think of how does a material deform on the surface, and is that influenced by hydrogen? And you know some of the results we've seen there is that it does change when you've got hydrogen, right? The amount of rotation you get towards those stable orientations is different in the absence and presence of hydrogen. We've never quantified that in more than one system. We've done it really in nickel thoroughly. There hasn't, at least I'm not aware of other systems where it's been done that thoroughly. So I think one place to start is pick other material systems and look and see if those evolutions of the surface strain, are they different? And that, because if you think of something that goes, I'm thinking to an experiment we did a long time ago where we actually fractured a sample of nickel with hydrogen inside an SEM, and you got the intergranular failure. But prior to that failure, you get incredible buckling of the grains.
Yes.
Which you never ever actually saw in the absence of hydrogen. We've never measured the evolution of that strain and compared it in the absence of presence of hydrogen, just on the surface. Does that also help give you an explanation of why there's a strain mismatch across the boundary? Now you're looking at strain mismatch, not microstructure mismatch. You're just looking at bulks, or strain as measured on the surface being different across grains. And does that depend on the grain boundary orientation? To me, that would be an interesting experiment that would say this strain incompatibility argument has some merit.
Yes. Okay.
But the nice thing is in these talks, I get to give all of you work and suggest future experiments for you to try rather than me doing it. So it's easy for me to think of the experiments to try, right? If I would certainly look at the strain evolution, even in a bulk sample. You have the techniques to do it now. 30 years ago, we didn't have those techniques. You do now. I would take advantage of them. The other thing I would urge you to do as a community, I think additive manufacturing gives you a unique opportunity to make specimens to get to the fundamental mechanisms. So rather than doing it in 316 steel or 304 or whatever titanium alloy you want to use, can you actually fabricate a system with a specimen geometry specifically to tackle a problem? And to me, I'd be looking at making clever sample geometries using additive techniques. You know, okay, it's got problems in microstructure, it's different, but I think you could design sample geometries that will allow you to uniquely explore some of these concepts, right? I think if you're really after the fundamental mechanism, that's where I would go. I mean, the steels that I used to produce, well, they're too hard. I think there's nice ways that you can make simple samples to do that. So
Yes, sure.
I'd encourage you to look at those kind of processes and how you could use those to make unique sample geometries and even compositions.
Yes, yes. Actually, we are doing some additive manufacturing stuff about the nickel. You will find that the nickel has an initial cell formation in already fabricated sample, and if we start from that point, maybe we can see further about the internal mechanism of these things. Yeah.
Yeah, the other concern I have with, if you've already got a cell structure, does that prevent the other cell structure from forming? So if you already dictated the cell structure that should form, and you don't give it enough time to evolve, that would be my only concern. But
yeah,
your experiments will tell you if that's a big problem or not.
Okay. Yeah. Thank you very much. Yeah. Thank you.
Is there any questions? Petrus, do you have questions? Before closing, do you want to say something?
Well, I'd like, on behalf of the entire I²CNER team and on behalf of our WPI friends from all over Japan and from all over the world, to thank Professor Robertson for this this challenging presentation. I think Ian, I enjoyed the way you presented the topics, the issues, and as you pointed out, the younger generations have a great a new collection of tools, and that they can investigate these issues and points that you brought up, and I hope they will prove that you are wrong. Okay. If they prove that you are wrong, then I'm wrong also. So we will both go down the history of having advocated wrong approaches. So it remains to be seen. But I think that's how science advances. I mean, you came into this international scene in the early 80s. I remember when I met you as a postdoc, right here in Champaign, Illinois, and you pushed the field further, and now it is time for the SWICE and our nodes and I²CNER to take over with the new approaches and techniques to push it even further. And I think this is exactly the WPI mission, right? The international cooperation and interaction. So I thank you for this challenging seminar that you gave. I think we couldn't have it better. Thank you very much. Thank you.
That was my pleasure. So only correction I'll give you, Petros, is that I think it's the students of Professor Kubota, of SWI, and the others, they're going to be the ones that solve the problem. I'll make this offer to the students this evening or this morning for you. Even though you didn't ask a question today, if you do have a question from the presentation, please send me an email. It may take me a little bit of time to get back to you, but I promise you I will eventually get back to you. But if you have some thoughts, some ideas, and you want to send me a note, and I'd be happy to try and help you and think of new ways of looking at this difficult problem. So thank you.
Okay, so it's time for closing. I thank Professor Robertson again. Thank you very much. I forgot to give you announcement. This lecture is recorded, and this lecture will be uploaded to the I²CNER web. So please visit I²CNER web page. So thank you very much, everyone, for joining this lecture. So that's all for this seminar. Thank you very much.
Well, thank you very much again. Thank you.
Yes.
Bye-bye.